Good morning, everyone. Thanks very much for joining. Myself, Camilla Humphries, and Doris Blaesing from RMCG. Unfortunately, we can't see Doris's face. We've just had some technical glitches, but today we'll be showing some slides, so it doesn't really matter. Our webinar will be brought to you by the Soil Wealth Integrated Crop Protection Project, funded by Hort Innovation and the Australian Vegetable and Melon levies. Doris will start today by talking about nutrition principles for vegetable production systems, and then I will follow by discussing nutrient use efficiency and budgeting. We will each be taking approximately 15 minutes, and we'll follow with a Q&A at the end. So please type your questions into the chat for us to respond to, and the webinar will go for approximately 45 minutes. We'll be recording this webinar, so if you have to go out into the paddock, you can view this later down the track. So thanks all for your attention, and carry away with Doris. I'll just share my slideshow. Oops. There we go.
Okay. So good morning, everybody, and thanks for joining the webinar. My name is Doris Blaesing, and I'm talking just a quick introduction refresher on vegetable crop nutrition, followed by Camilla. Camilla, you will probably have to change the slides because you're opening up the presentation. So can you show the next one, please?
Certainly.
So as I said, a quick nutrient refresher, and then looking at the factors affecting nutrient uptake and how we can actually measure and monitor and plan better. Next one, please. I put this reminder there that, there actually are 15 essential elements, and 3 of them are carbon, oxygen, and hydrogen. They're actually the major building blocks of plant matter. So we mustn't forget that. And then there are 12 major and minor nutrients or trace elements, and they're all equally important. It seems still a lot of people have a focus on NPK, but in vegetable production, especially calcium and sulfur are really important. Sulfur is often forgotten, and if people don't use sulfate forms of fertilizers, we find sulfur deficiencies who very much can look like nitrogen deficiencies. So we're not actually looking at any deficiency symptoms today, because once you see them, the crop is already damaged. And one important aspect is that a lot of the nutrients can actually supplied by organic matter or organic amendments. So we just briefly want to touch on that as well. Probably everybody of you has seen this barrel showing, the effect of a limiting factor. So the yield gets affected by a limiting factor, and usually that's used for plant nutrition. But obviously, with the drought conditions, water is obviously very limiting factors, but soil conditions are limiting. And quite often, especially when we get computer-generated recommendations based on the soil test, we forget about soil conditions. And definitely poor soil structure and salinity can be major limiting factors that we have to deal with. And while salinity can be dealt with, for instance, with good nutrition, poor soil structure cannot. Next one, please. So what are the key limiting factors other than nutrition? Loss of organic carbon is an issue. It's a huge driver of yield and often neglected. That soils is heavily tilled quite often in vegetable production, and once organic carbon goes below kind of the natural level of the soil and isn't replenished by either amendments or cover crops and crop residues, the crop will be far more difficult to manage, and nutrition will be suffering. Soil biology just goes along with organic carbon, and we have to we try to measure it, we try to understand it, but still the main indicator for good biology is probably still, we still haven't found which indicator to measure and which the corrective actions are. But I can talk a bit more about the importance of soil biology. Soil structure is hardly ever measured. It's quite complicated to measure, but I will just bring a few indicators of how you can look at soil structure and how that should be considered when you look at nutrition. Especially, I think, in the dry conditions, we get more issues with salinity, so high levels of sodium chloride in either the water or the soil. Once it's in the water, it's obviously also in the soil. Some areas are affected by sodicity, which is just high sodium without the chloride, which has a really detrimental effect on soil structure. It often goes along with high magnesium levels. The soil gets really sticky, get really hard when it's dry, and nutrition is quite difficult to manage. So there are some methods that can be managed through nutrition, especially calcium and potassium. However, we also need to add organic matter to actually reduce the effect of salinity and sodicity. When we look at soil sampling, often only the top 10 centimeters are sampled, and we don't really look at subsoil. And I think it should be done, especially in new paddocks, but once in a while we should look deeper than just 10 or even 30 centimeters and check any constraints. Acidification, low pH drop is quite normal, and usually in vegetable production, lime is applied to counteract, acid conditions. Sometimes people forget that, for instance, gypsum does not change pH, and that urea or any ammonium fertilizers acidify the soil quite strongly. So we need to consider that when we manage nutrition. I have on the top right, there's an image of a NDVI image, and it shows really the different soil conditions that can appear within the paddock. So if we use the good old methods of zigzag sampling, we might get an answer that doesn't really help us with good nutrition. So it is worthwhile having either NDVI, even a more historic image, or using Google Earth to see whether we can zone the paddocks and take representative samples of certain areas rather than a mix of everything, so that nutrition doesn't, we have the average nutrition, which never is the optimum. One of my favorite photos is a bottom right hand, a really good root system where you can see the soil adhering to roots, giving a really good rhizosphere, which is essential for good nutrition efficiency. And Camilla will talk a bit more about nutrient use efficiency. Can you please show the next one? And the sticks, yep. So what we usually check are the fertility indicators. I guess hopefully every agronomist and grower does that. So taking a normal soil test, hopefully taking a subsoil test as well. So you learn about nutrient levels, balances, EC, pH, organic carbon, which are really important indicators. So next one. And I talked about soil structure and texture, really important to know and check. Texture is just the coarseness or the fineness of the soil, but it's a clay, sand, sandy loam. But structure means, how many holes are in for drainage, how much air is in the soil, which is really important. People forget about air all the time. Next one, please. And soil life. So that's the great unknown. We're still learning about soil life, but there are some measurements and some things we are learning, and I very briefly will talk about that. Next one, please. So if you go to soil texture and structure, so there are things you can test. Penetration resistance is interesting. It's very much influenced by water, so you can't test one day here, another day there, and compare the data. But what you can do is, if you look at different treatments or different management aspects within a crop, penetration resistance can give you really good data. You can look at aggregate sizes, and I have another slides where I show you how to do that. That's an in-field assessment. You can look at aggregate stability. If you want to build routes, that's done in the lab, but you can actually press the aggregates and see whether they're easily crumble or not. So they should easily crumble, which is good. Dispersion, there's an image on the right showing different dispersion. If dispersion is high, there's an issue with solidity and calcium is needed. For instance, slaking is number 6. Slaking shows how the aggregates fall to bits, and if they slake easily, there is an organic matter requirement. One really good one to look at structure is actually drainage and look at infiltration rate. So you could even use a piece of downpipe, fill a certain amount of water in, and measure how quickly it infiltrates the soil. If it's very slow, you're going to have a problem with irrigation, actually getting the water in, but also getting drainage in the root zone. The soil will dry out quickly and it will wet quickly. And that has a huge effect on nutrition. And then, of course, soil texture has an influence on nutrient holding. Sand doesn't hold much, clay holds much more. The cation exchange capacity is the indicator used there. And organic matter has got the highest CSC and the highest nutrient holding capacity. Next one, please. So one of the really important, easy things, of course, to do is to use a visual soil assessment. And just thinking that soil structure can actually have a much greater effect on nutrient uptake and the crop than actually the amount of fertilizer used. So it's very important to have friable soil. And one of the Tasmanian soil scientists developed this soil structure scorecard on the right, which is actually fantastic. We could, refer to the scorecard and actually say, well, a potato crop will have only 65 or 85% of yield, depending on the structure score. So that's a really good instrument if you don't want to do lab measurements, to actually take a spade and have a look. Next one, please. Next, so we go to soil life. People often talk about what's here listed as microflora or microbes. The really important thing to remember is that we need the macrofauna, the mesofauna, and the microfauna to actually initially break down organic matter. And the good thing about especially the macrofauna and the mesofauna is that you can actually see their activities. If you have, soil clumps in your hand, you can see whether there's little holes in there, which means there is some life in the soil actually starting to work and starting to break down organic matter. Earthworms will kind of pull organic matter deeper into the soil. Earthworms actually can have more power than roots to penetrate soil. So that's a good thing to know. But just remember, it's not just the microbes which are measured in labs, bacteria, fungi, viruses, and also part of the microflora are all our lovely soil-borne diseases. So they're part of the whole mix. And often research looks at them separately, whereas I think it's about time to look at the pathogens and the good guys together. Next one, please. So there's some test examples that can be used to look at microbial activity in the soil. A really good one is actually they use cotton strips and see how quickly they are decomposed. You might remember that Soil Your Undies initiative, where people actually used cotton underwear and did a competition amongst different growers of whose underwear breaks down more quickly, and the more quickly it breaks down, the better the soil. But you can actually use just a bit of cotton. Next one, please. Talking about soil biology, the question is always asked, do biological products work? And the answer is it depends. There are some really great biological products. Some of them are registered with the APBMI. Others might be more in the snagger category. But the big message is that the biologicals are not going to fix really poor soil conditions. So you need to really use them as the icing on the cake and have the basics right, soil structure right, nutrition have to be balanced, and the water has to be right. Without all that, there's even with a very good biological product, you can't get a big effect. So the next one, please. With this one, actually, one of the questions again about biologicals and soil amendments is to do their work. Does organic matter work? And organic matter is probably what I call black magic. It does actually help with biological activity. It helps with soil structure, water. It provides food. It holds onto nutrients. So the more you can do to maintain organic matter and to increase it, the better. If your soil test result gives you organic carbon, you multiply that by 2, and then you know how much organic matter you have. So that's a pretty simple relationship. And I just want to highlight the kind of citation there from Wendy Seabrook, that we have not provided a good catering service to our soil life. So again, they do like organic matter, and they do like a good buffer with different types of organic matter, for instance, cover crops. And Camilla will talk about that a bit as well. Next one, please. Then we come to the actual nutrition consideration. So we've looked at our soil, we make sure the structure is all right, now the water is going well, there's enough organic matter, the sea is good, or we know we need to watch salinity. So what else do we then consider? One major consideration is growth stages. And quite often, a lot of fertilizer is actually applied already early on as a starter fertilizer, and really small seedlings, can't really use that fertilizer, and especially nitrogen or sulfur or boron might be lost by the time it's actually needed. So it's probably worthwhile rethinking, crop stages and spreading the fertilizer program according to crop stages. Another thing is to actually understand removal rates. So really, the amount of nutrients applied to a balanced soil should be what the crop would remove. So there are lookup tables like this one, so you could see what broccoli removes or what red cabbage removes, and then you can budget accordingly. So for instance, in Germany, where I come from, you're actually not allowed by law to add more nutrients to the soil than the crop you want to grow removes. That's pretty challenging, because sometimes it's better to have a bit of a buffer there. But the main thing I want to point out, for instance, when you look at potassium and nitrogen, the removal rates are very similar. And still, potassium sometimes is neglected as a nutrient that actually has high need. And it's really important for fruit development, for root development, and for water regulation. So plants who don't have enough potassium will get stressed far more easily and lose water through the stomata easily. Celery has a huge potassium need. Beetroot has quite a big one. So it's really worthwhile, getting these lookup tables for nutrition needs and removal rates. The next one. Here's just one slide. Maybe you want to photograph it, or we also have it available in other publications. Is that at different growth stages, some nutrients are needed more than others. So, and I highlighted, potassium is not that important early on, but certainly once plants are growing and really powering, you need the potassium. Even if there's a reasonable amount in the soil, you could still support growth, rapid growth through more potassium, and foliar applications do work. And probably the other ones is looking at sulfur and micronutrients. So the Soil Wealth project has published and keeps publishing some nutrient management guides for different crops, and this table will occur in this guide, so you can look at the guides and get more information for certain crops on what is needed when. So one example is the next slide from the Brassica nutrition guide, which actually shows the crop stages, the nutrients required, and what the focus should be. So that's quite a helpful reminder on when you're planning a nutrition program, and which fertilizers to use and how much to use over time for the different crops. Next one. And I guess everybody knows the right source, right time, right place. And the right source is sometimes quite difficult to determine, because there are a lot of different types of fertilizers. And generally for vegetable production, it's good to use some with low salinity index, low acidifying potential, which excludes urea and ammonium sulfate really. and don't use chlorides really, because plants need very little of chloride. So that's about the right source. Compound fertilizers, slow release fertilizers are really good to run a program the right way. We talked about that, crop removal, understanding conditions like salinity, where you might need more potassium and calcium, timing, timing to crop stage rather than giving big hits at any one time, and placement. Bend placement can be good for phosphorus in heavier soils, or foliar applications could be the right place actually to apply. And then my last thing is to actually monitor the crop to manage it. So I think still sampling of crops is not done often enough. It's an additional cost, but the growers are doing it, get their money back big time. So this is just a summary of what we talked about. Check soil condition. One thing I would personally never do is to give fertilizer recommendations without actually having checked the paddock and understanding that, and understanding also the equipment and the capability, what's actually there to use fertilizer. So how can we do the right thing for the right crop, the right soil, and the right farm? Soil tests, hopefully everybody is doing routinely. Some people don't do it that often and get some weird outcomes. So the soil test certainly pays for itself. We talked about removal rates, crop stages, and a balance of nutrients. Especially overfeeding of nitrogen can be a real problem also for pest and disease management. We talked about the right fertilizer and application method, and then the timing as well. So again, step 7, monitor and adjust. Don't wait until you see deficiency symptoms, because that means the crop is already damaged. And usually insect pests and diseases are very good at recognizing crops that are not well fertilized, and they'll latch onto them first. So that brings my part to an end, and now we went very quickly through certain things. The Soil Wealth project has a lot of further information on what we just talked about, and webinars on soil test interpretation, booklets on soil test interpretation. So have a look around the website, or give me a call if you want to know more. Thank you.
Thank you very much, Doris. Doris has covered a lot of the principles, and now I'm going to instill some of these principles in how to improve nutrient use efficiency through nutrient budgeting and monitoring. So first of all, what is nutrient use efficiency? It refers to how well a crop uses nutrients from the soil and applied fertilizers and converts these to growth and yield. So nutrient use efficiency is influenced by a number of factors, and they include plant genetics, soil properties, climate, and agricultural practices, particularly soil management, irrigation, and nutrition from soil and soil amendments and fertilizers. So we're using nitrogen as an example of nutrient cycling, as it is a particularly dynamic nutrient. And this figure shows nitrogen uptake, cycling, and recycling in a vegetable production system. And it shows that nitrogen comes in multiple forms and sources. Mineral fertilizers are taken up by the crop. Nutrients are then recycled to the soil via crop residues and then cycled through the ecosystem by microorganisms, which mineralize the nutrients into plant available forms of ammonium and nitrate. The biological processes in the soil are influenced by soil pH, humidity, soil moisture, oxygen, and temperature. So nitrogen cycling is disrupted when plants or organisms are removed and nutrients are lost via leaching through the soil or volatilized into the atmosphere. So just to bear in mind that nitrogen fertilizer is usually used as a top-up to the nitrogen which is already in the soil. So it's really fundamental to do baseline soil nitrogen tests. Now we're referring to all nutrients, and nutrient uptake in crop cycling is continuous. The amount of nutrient must match the crop growth stage, as Doris has mentioned. it is really important to apply the right source at the right rate at the right time in the crop's growth stage to ensure nutrient uptake is effective and efficient, and to avoid excessive fertiliser use, which can be very expensive, and also to avoid nutrient leaching through the root zone. So why do we consider soil amendments in a nutrient budget? Soil amendments or conditioners have a significant impact on nutrient use and availability. So therefore, we need to consider soil amendments when we do our nutrient budgeting. Soil amendments can provide the following benefits: better soil structure and stability, better water holding capacity and drainage, provide food for microbes and invertebrates, which then break down nutrients into available forms and organic matter to hold onto more nutrients on the soil surface. They also improve root growth by providing more air spaces and improving porosity for roots to grow and take up more nutrient from the soil. So this image here is of a typical Australian clay duplex soil, which shows layering and compaction between the A and B horizons, which dramatically restricts root growth. So ameliorating the subsoil, as Doris mentioned earlier, the first 30 centimetres with an organic amendment can significantly help improve soil structure and root penetration to access those nutrients. So this table I wanted to show you is comparing different organic amendments. It's been extracted from the Gippsland Deeper Soils project, which has been led by Bill Grant from Federation University, which shows that one organic amendment is not like the other. Compost does not have much nutrient. However, manure is much higher in nutrient content. The project aims were to deepen the topsoil by integrating different soil amendments in the top 30 centimetres. And the best performing amendment was the matured municipal compost, so seen in this first column on the left there, displaying different physical, chemical and biological attributes compared with other commonly used soil amendments. So they included better pH buffering, improved and higher cation exchange capacity, improved water holding and availability, and better soil conditioning and improved physical porosity of the soil, as well as promoted soil biology, so better fungal and bacterial count. Just before using organic amendments, it's important, like composts, it's important that you ensure that they are AS4454 certified, or get the specifications from your composter, just to ensure that you are using quality product. Cover crops are part of the nutrient cycle. Everything in the system has to be accounted for. So cover crops are considered a catch crop for residual nutrients left in the production system post-harvest, which can then be cycled back for the next crop. The removed nutrients, such as nitrogen, can be supplemented by the incorporation of particular cover crops, especially nitrogen-fixing legumes, such as lab, cowpeas, chickpeas, sun hemp, clover, lupins, and vetch. For nitrogen, it is really important to use the correct rhizobium inoculant to achieve good nodulation. And you need to know the right rate of rhizobium inoculant levels to apply in order to achieve this. So legumes fix 100 to 200 kilograms of nitrogen per hectare, and they won't fix much nitrogen if your soil nitrate levels are above 100 kilos of nitrogen per hectare. And just to bear in mind, for heavily compacted soils or soils in poor condition, nitrogen fixation won't be achieved. The Soil Wealth integrated crop Protection Project demonstration trial, shown here at Butler Market Gardens in Skye, Victoria, has been trialling cover crops to improve nitrogen cycling and the management of nitrogen inputs. And the case study has been written up and is now available for you to read on our website. The three different treatments of cover crops we tested included mixed legume species of lab, cowpea, soybean and sun hemp, compared to buckwheat and vetch, which was then compared to sorghum. And sorghum produced the highest above ground biomass, and its nitrogen uptake for recycling was the highest of all the cover crop types. This photo here is of me in the buckwheat cover crop just prior to termination. And it's vital that you have a termination plan, as the method of termination and timing is particularly important when you're trying to really break down that biomass and cycle it back into the system, and also to prevent weeds. Cover crops can also help with other things like soil structure, as Doris was really emphasising before. High root activity stabilises the soil and encourages mycorrhizal growth. There are also particular cover crops that can be used as biofumigants, and they impart glucosinolates when macerated and can help break down the pathogen fungi in the soil. They can also be used for weed control, but sown at a higher rate, roughly 30% higher than normal, to outcompete weeds. But bear in mind that buckwheat can become very weedy if you don't terminate it at the milking stage. So timing is important. There are a lot of other cover crops trials and information on the Soil Wealth website, because we have been, or the project has been doing this work for the last 11 years. So there's a very useful cover crop termination guide that can be accessed on the website too. So just going on to the benefits of controlled release fertilisers for plant available nutrients. They can really help make nutrients available throughout the crop cycle. There are different types of slow release fertilisers, biodegradable polymers, which contains a compound fertiliser, NPK, within a gel coating, and that is then broken down upon wetting. They're nitrogen inhibitors, so that slows the conversion of ammonium to nitrate to reduce nitrous oxide emissions. Also urease inhibitors, which slows the conversion from urease to ammonium, and then biofertilizers, so these can be derived from chicken manure, and activates carbonates in the soil to stimulate microbial activity. So this graph here over to the right, I conducted a pot trial, a leaching trial, showing the rate of leaching for different fertilisers compared to the polymer-coated slow-release fertiliser. The x-axis shows time as number of leaching days, and the y-axis shows electrical conductivity. So the slow-release fertiliser, shown here with the orange line, plateaus below an electrical conductivity of 3, with the other fertilisers, the grey and blue lines, rapidly peaking at 5 within the first 2 leachings. So it just goes to show that the slow-release fertiliser is more efficient and prolongs the release of those plant essential nutrients to the plant over time. So reducing the level of leaching. So we are currently conducting a controlled release fertiliser trial in the ground at the Skye demo site on their current coriander crop. to demonstrate the release of plant available nutrients over the crop cycle. So as part of this trial, we're using the Centex Solu samplers, which are inserted into the root zone of the crop, and they take up plant water via capillary action, and the water is extracted from the tube, and then the nutrient solution is then measured to identify which nutrients are present and at what levels in the leachate. So when forecasting for nutrient removal, you should refer to published data on nutrient removal rates, and Doris showed in her presentation some data on nutrient removal rates for various vegetable crops. But when you're trying to work this out yourself, it's important to take a baseline soil test and monitor the crop during the life cycle, and at harvest take a representative dry tissue test of removed biomass and calculate harvested dry weight as kilograms per hectare. So using that formula highlighted there, and all tissue which is not harvested then goes back into circulation. Then based on those results, you then replace the percentage nutrients removed through either organic amendments and baseline and side dress fertilizers. So one example is here for a broccoli crop. Nutrient removal is the amount of all nutrients removed at harvest in kilograms per tonne of dry matter. So here to calculate the removal, we've got approximately 60,000 heads per hectare planted. We've come up with an average per head weight harvested to come up with the total kilograms removed from the paddock, calculating the dry matter removed, 3,600 kilograms, and then tested for nitrogen specifically, where 4.5% nitrogen was found in the dry matter heads in the dry tissue tests, to come with 162 kilograms of nitrogen removed at harvest. It's just interesting to note here that the potassium removal rate is as high as the nitrogen removal rate. So as Doris mentioned earlier, it's particularly important to bear in mind different removal rates of different elements, but that potassium one is very, very high. So for further information and further reading based on the two presentations by Doris and myself, you can go and have a look at some calculators, conversion tables, and the Soil Wealth project has published rules of thumb poster for nitrogen from compost and organic matter. And we've also published a fact sheet on nitrogen cycling and accounting for increased nitrogen use efficiency. So just going to spend the next 10 minutes or so answering any questions you have. And there are contact details if you would prefer to speak to us individually or out in the paddock sometime for one of our Soil Wealth field days. Thanks very much for your attention.